Okay, we're going to continue on with some of the chemical agents that are popular for use for controlling microbes. The next category are the phenols or phenolic compounds. And so this is a diagram over here which is showing you the chemical structure of phenol or phenol, different pronunciations. This is an organic chemical, and remember when you see these types of chemical diagrams, it's understood that there are carbons at the points there, and those carbons are bonded to hydrogens, and they're just not drawing all that in for simplicity. So that's phenol or phenol. Now this was the first, historically, this was the first chemical agent that was used as an antiseptic, the first use in antisepsis for surgery, and it was Joseph Lister back in the 1800s. So back then, they did surgery on people, amputated legs, cut them open for various things. And, generally kind of what happened is you had your surgery and you got some horrible infection from it and you probably died. That's kind of the way surgery went back then. Joseph Lister came up with this idea of, hey, well, while we're doing surgery and we have open incisions, let's spray this chemical called phenol over the incision and see whether or not that makes a difference in survival and health of the patients. And sure enough, it did. It cut down on infections after surgery. And interestingly, these discoveries were made before we fully understood that microorganisms were responsible for human disease. So they just realized that this phenol stuff was working and helping the patients, and they didn't really understand fully the reasons why. But anyway, this was the original chemical antiseptic used in human health. And yes, good old Listerine over here is named for Joseph Lister. And all right, so there are some various derivatives of phenol. These are called phenolic compounds. So they all have this basic structure, and some of them have two of those chemical ring structures bonded together. And some of these are found in, they're found in various things that you use. Mouthwashes have phenolic compounds in them. Now this one, triclosan, I want to draw your attention to because this is most things that are labeled antibacterial have triclosan in it, and they're putting that stuff in everything. Your antibacterial soaps and dishwashing detergents, and they even put it in trash bags and things like that. And in fact, it's being used so much that, hey, we just learned about how bacteria and other types of microorganisms are good at mutating. And they can become resistant to things that we expose them to. And that's, sure enough, that's what's happening. There's a lot of triclosan resistance developing in bacteria. And, because of natural selection, the more we keep using triclosan, the bacteria that are sensitive to it die off. Think about it. All this stuff gets washed down the sink. It's in the water supply. And so out in open waterways and so forth, you're just selecting for microorganisms that are resistant to triclosan, and pretty soon all these nifty antibacterial soaps and dishwashing liquids and diapers and so forth not working too well anymore. So that's a concern. These are generally intermediate to low-level disinfectants. A lot of natural disinfectants have naturally occurring phenolic compounds in them. So, things like pine oil and clove oil, many of these oil-type things that people use as natural antiseptics are really, they contain phenolic type compounds. Interestingly, some of these are active against that mycolic acid cell wall of the mycobacteria, like Mycobacterium tuberculosis, which we've talked about how difficult of an organism that is to control. These are pretty good at disrupting cell membranes, breaking cell membranes apart, and denaturing proteins. Some of them are irritating, though, like chloroseptic. Remember, if you've ever had that, use that as an antiseptic for a sore throat, and you spray that, that nasty taste kind of burns the back of the throat, and that's because that has a phenolic compound in it as an active ingredient. Chlorhexidine, this is a pretty commonly used for scrubbing, degerming, and as an antiseptic in medical facilities. And so it's kind of a combination between a phenolic compound and a chlorine-containing compound. So it's a complex molecule with phenolic chemical rings and chlorine atoms. Works, it disrupts cell membranes, denatures proteins with long enough exposure time. Hibiclens, if you've ever used that, pink stuff, is kind of commonly used as an antiseptic soap when you've got to really try to knock down the numbers of microbes on the skin. So this is used for prepping skin for surgery, sometimes used in irrigation of mucous membranes. We're trying to flush off a mucous membrane surface. Pretty good stuff. It's low toxicity. It's mild. You're not likely to suffer any effects from it. Doesn't kill everything. It's not a sterilizing agent. Pretty good against various microorganisms, but not all of them. There's a lot of variability with it. All right, how about good old alcohols? All right, so alcohols include things like rubbing alcohol, that's isopropyl or alcohol or isopropanol, ethyl alcohol or ethanol. And that's the type of alcohol in your beer and your wine and your liquor and so forth. That's your drinking alcohol as well. So alcohols are generally classified as intermediate level disinfectants. So they're not going to be good at killing bacterial endospores. They're not very good at killing fungal spores either. So just keep in mind, when you use alcohol on the skin or a surface, it's not killing everything. You're not achieving sterility. This tends to disrupt cytoplasmic membranes and denatures proteins and dehydrates cells as well. Kind of a downside of alcohols is they dry really rapidly. So you guys know that. Alcohol evaporates really fast, so you don't have long contact time, so that's going to help limit the effectiveness of using these. But nevertheless, they're still pretty good. They're commonly used as everyday antiseptics and disinfectants. I've already mentioned that if you have other chemicals, antimicrobials, that are dissolved in alcohols, those are called tinctures. Oh, hand sanitizer. You know that stuff that everybody uses, pumps out all the time, rubs on their hands? The main active ingredient in that is ethyl alcohol. Those are alcohol-based products, and that's how they wind up controlling microbes on the skin. I would caution you guys, though, we kind of live in we'll talk a little bit more about this later, especially as we get into the immune system. We live in this overly hygienic society where everybody's paranoid about microorganisms, and we got to murder all of the germs and get rid of them. And as we move beyond here and into chapter 11 and 12 and 13 and so forth, we want to think about whether that's really a good thing to do. And even talking here about how microbes become, the more we expose the microbial world to all of these chemical control agents that we have, the more likely they are to mutate and become resistant to them. And then those resistant members become the dominant members of their population. So we have to think about whether this sort of widespread, constant use of all this antimicrobial stuff is necessarily a good thing overall for human health and the environment. All right, detergents and surfactants. So these are kind of in a category together, and these are molecules that have a hydrophobic portion and a hydrophilic portion of the molecule. And so hopefully that kind of reminds you of phospholipids that make up cell membranes. That's what they're trying to show you over here. So here's a cell membrane, and in blue here, the square portion, that's supposed to be a surfactant molecule, and the square portion is supposed to be hydrophilic. The stick portion there is hydrophobic. So the hydrophobic portions will love those fatty acid tails of the phospholipids in the cell membrane. So they're going to wiggle their way down in there, and that's going to help break apart the cell membrane. So that's generally how things in this category work. So any detergents that you use, basic soaps and so forth, they work through disruption of cell membranes for the most part. All right, now there's a category of surfactants called quaternary ammonium compounds. The abbreviation for that is QUATs. These are very commonly used. They're used a lot. Some of those are up in the intermediate level category for how effective they are. And quaternary ammonium compounds, they have chemical structures that look here, a couple of examples over here. Cetylpyridinium and benzalkonium are a couple of examples of QUATs. They're called quaternary ammonium compounds. This is what an ammonium ion looks like. It has a nitrogen in the center bonded to 4 hydrogens and a positive charge. And so they all contain that basic general structure somewhere in the molecule. And they'll have a hydrophilic portion around that positively charged nitrogen. Things that have positive and negative charges love water, hydrophilic. And then they have a long tail of carbons and hydrogens, and that's hydrophobic. That doesn't want to interact with water. So like we were taking a look at over here, that makes those molecules really good at breaking apart cell membranes. These can actually be included in mouthwashes. So they sound kind of serious because, but they're heavily diluted. You know, they're down in the parts per million range for effectiveness. Because you want to find a concentration, if you're putting this in mouthwash, something that's going to break apart the membranes of bacteria that are in the mouth and help limit the numbers of microbes that are there. But you don't want this damaging or disrupting your own cell membranes or on your own tissues in the body. So you have to be kind of careful with the concentrations and exposure times. That's why they tell you to, if you're going to use mouthwash, you don't gargle for like 30 minutes because you don't want to overexpose your own tissues to these chemical substances. You keep those exposure times down to try to target the microbes and keep your own living tissues safe. Heavy metal. These include things like arsenic and zinc and mercury and silver and copper. So this is there's certain elements on the chemical periodic table that are classified as heavy metals. And these two have antimicrobial properties. They do that by attaching to the amino acids in proteins. Remember, one of the ways that proteins can get screwed up is that other chemical substances can attach to them and say maybe block the active sites of enzymes. So these either alter the shape or block active sites, and that's how they function. Most of these are pretty low level. A lot of these are toxic to us as well, so you have to use them at extremely low concentrations. So they're not going to wipe out things like endospores. Endospores are going to be resistant to this. Mercury, that was included in, if you're old enough, you might remember a medicine, an antiseptic called mercurochrome. It had an orange color. I know my mom loved that stuff, and hydrogen peroxide as well. And the antimicrobial properties in that came from very low amounts of mercury that were present. Copper can be used as an antimicrobial. They'll actually include copper now in surfaces, in operating rooms and so forth, because microorganisms are less likely to grow on copper-containing surfaces. Copper gets put. Some people use copper to help control the growth of fungi on vegetable plants in their gardens. Silver nitrate drops in the past were put into the eyes of newborns. The idea there was if the mother had gonorrhea, if she was infected with Neisseria gonorrhea, those silver nitrate drops could prevent that bacteria from getting into the eyes of the baby during delivery. And Neisseria gonorrhea infection of the eyes in the newborn can cause blindness. So that's where that practice came from. Now they've switched over and they're putting antibiotic drugs into the eyes of a baby when it's born to help prevent eye infections. All right, I've already talked about this, but I want to harp on it a little bit again. Are we forcing evolution of microbes resistant to antimicrobial agents? So we've talked about mutations. So microbes they reproduce a lot, so they're replicating their DNA a lot. The more you replicate your DNA, the more of a chance there is that some DNA mutation can come along that makes a individual microbe resistant to something that you're exposing it to that's supposed to kill it. And if it's resistant, it's going to kind of have like a superpower now, and it's going to be more likely to survive and reproduce and pass this genetic mutation that made it resistant onto its offspring in subsequent generations. And so that's an example of evolution on the small scale, natural selection at work. The more that we use disinfectants and antiseptics, antibiotic drugs, which we'll be talking about as we get into chapter 10, the more that we use these, the more that we're pressuring these microbes to mutate and become resistant to them, and the more those resistant populations begin to dominate. So we have to be worried about these resistant strains that are out there. And already we're seeing resistant strains of some bacterial species that are very, important pathogens. Mycobacterium tuberculosis, Pseudomonas aeruginosa, Escherichia coli, Staphylococcus aureus. There are strains of these that have become resistant to some of the common disinfectants and antiseptics that are used. And so once that happens, then you got to find other things to use to try to control those types of microorganisms. So it's pretty serious stuff to think about moving forward. Take a look at the table in your book, which just shows you for some pretty common commercially available antimicrobial chemicals out there. It's kind of interesting just to take a look at this and see the specific chemical agent, that's your active ingredient, that's present in each of those, and then what category they fall into. Is it a detergent or a quat, halogen, phenolic, alcohol, etc.? That's pretty interesting to take a look at. Kind of last thing, I wanted to point out something from a cultural perspective. If I ask you to think briefly about cuisine, foods around the world from hot tropical places, so think about like Mexico, Central America, think about the southern parts of Asia, like Vietnam and Thailand and India, and even into parts of Africa. What would you think about those cuisines? Maybe you're thinking they're very spicy. A lot of times they're very hot, intensely flavorful. And so why is that? Why is there this correlation out there between the types of cuisines that are favored among the populations of people who live there? Why has that happened? So there's a idea out there, a hypothesis that's out there, that populations of people that lived in hot tropical regions developed through natural selection. They developed a taste for very hot, spicy foods. And the reason for that is many of these intensely hot, spicy foods have the spices. There are chemicals in the spices that help prevent microbial growth. So, if you're living in a hot tropical area and you can't refrigerate your foods, they would be less likely to spoil. If there are pathogens in those foods, those pathogens would be less likely to be able to grow up to numbers that would be harmful if you have these spices and chemicals in those foods that are preventing the growth of microorganisms. So that's I've actually got a link here in the PowerPoint that you can click on the full version of the PowerPoint on Blackboard if you want to read more about that. It's kind of an interesting hypothesis that it was actually evolution, natural selection that drove these populations of people in the hot tropical parts of the world to, over time, develop this preference for these hot, spicy foods, because those hot, spicy foods contain things that have natural antimicrobial properties. I've got garlic up here because garlic is a very good antibacterial. And just kind of as an example, there's a petri dish here on a plate, and it has been spread with what we call a lawn of bacteria. So it's supposed to have bacteria growing like one big massive colony across the entire plate. And right there's a little paper disc that was soaked in garlic juice. I've got it labeled there with a G. And you notice there's a big clear zone around that paper disc. That's called a zone of inhibition. So there are chemical compounds in that garlic extract that diffuse out into the agar, and they prevent this bacterial organism that's in the Bacillus genus from growing anywhere near that disc. So garlic has some pretty good antibacterial properties. These pictures came from a salsa experiment. There were some students who were testing, not here at Calhoun, but they were testing extracts of various things like tomatoes and cilantro and jalapeno peppers and so forth for antimicrobial properties. And it's interesting, there are a lot of naturally containing compounds in herbs and other types of foods that have pretty good antimicrobial capabilities. So, and garlic is good stuff. Not only is it a good antimicrobial, but it also has very positive effects, like for your cardiovascular health as well. And it's also been studied for anti-cancer properties also. So eat plenty of garlic, just don't breathe on anybody. All right, that actually concludes Chapter 9. Now we're going to roll right on into Chapter 10, where we're going to be focusing more on antimicrobial treatment of patients. So not so much rubbing an antiseptic on the skin or gargling with mouthwash, but instead drugs, things like penicillin that you take and spread throughout the body to try to kill microorganisms that are causing an infection. So these are things that you're going to use to help enhance your body's natural ability to fight off infection. So that'll be the subject of Chapter 10.